everyone, I am Ruzelle Mikaella, a Taib. I am 17 years old and a grade 12 student of STEM 1207 St. Maria Gorete from Notre Dame of the Jangas University. Today, we will be making a 3D bacterial cell for our midterm performance test in the subject of General Biology 1. Later on, I will explain to you and present the specific structure and functions of the cell and the organelle. So, what are you waiting for? Let's get started.
The materials that we need are scissors or cutter, cartolina or construction paper, cartoon or cardboard, glue gun and stick glue, yarn, foam sheet, beads, and paper clip. First, we need to make a base or a shape of a bacterial cell using a cartoon or cardboard. I also use foam sheet for the base of my bacterial cell to be thicker, but it's up to you what to use and how your base will be thick. Next, I will be layering all the carton that I have cut earlier. I will be covering the side of the cartoon using the cartolina, and for the front and the back, I will be using construction paper. Then I will use red yarn for the plasma membrane, purple yarn for the DNA, beads for the ribosomes, paper clip for the tail that represents a flagellum, red yarn again for the plasmid, and black yarn for the pilus.
I will explain to you the functions of the different parts of the bacterial cell. So, let's get started. The first part is the cell wall. This color pink one is the cell wall. Here. The cell wall surrounds the plasma membrane of plant cells and provides tensile strength and protection against the mechanical and osmotic stress. It also allows to develop turgor pressure, which is the pressure of the cell contents against the cell wall. Next is the capsule. This color green one is the capsule. The capsule can protect a bacterial cell from ingestion and destruction by white blood cells or phagocytosis. While the exact mechanism for escaping phagocytosis is unclear, it may occur because capsules make bacterial surface components more slippery, helping the bacterium to escape engulfment by phagocytic cells. Next is the plasma membrane. This red one here is the plasma membrane. The plasma membrane or the cell membrane provides protection for a cell. It also provides a fixed environmental inside the cell. And that membrane has several different functions. One is to transport nutrients into the cell and also to transport toxic substances out of the cell. The next part is the nucleus. Here, the violet one in the center is the nucleus. The nucleus is essential for controlling the activity of the cell and reproduction. It is where transcription and replication of DNA take place. Next is the ribosomes. These are the ribosomes. These many dots, these are the ribosomes. Ribosomes have two main functions: decoding the message and the formation of peptide bonds. These two activities reside in two large ribonucleoprotein particles or RNPs of an equal size. The ribosomal subunits. Each subunit is made of one or more ribosomal RNAs and many ribosomal proteins or R proteins. The sixth part is the cytoplasm. This blue is the cytoplasm. The cytoplasm is the gel-like fluid inside the cell. It is the medium for chemical reaction. It provides a platform upon which other organelles can operate within the cell. All of the functions for cell expansion, growth, and replications are carried out in the cytoplasm of a cell. The seventh part is the pili. These black here are the pilis. The pilis are short, hair-like structures on the cell surface of prokaryotic cells. They can have a role in movement, but are more often involved in adherence to surfaces, which facilitates infection and is a key virulence characteristic. The eighth part is the plasmid. This one. They are the DNA, helps in the survival of the organism. It kills other host cells by producing toxic proteins and also protects its own cells by developing resistance or killing the foreign organism. The plasmid also helps in replicating the bacterial DNA in some harsh conditions. This tail is the flagellum. The flagellum is primarily a motility organelle that enables movement and chemotaxis. Bacteria can have one flagellum or several, and they can be either polar, one or several flagella at one spot, or peritrichous, or several flagella all over the bacterium. Hello everyone, I hope you're doing fine today. Time check, 3:45 in the afternoon. I am happy because I already finished making the 3D diorama of my bacterial cell. I'm also happy because I didn't spend even a peso on it, just for this project in the General Biology 1 for the 3D diorama, because all the materials I used for it came from my past performance tasks. I hope you've learned something in our video. Thank you for watching. Bye-bye!